All right. So this lecture is about molecular representations and core descriptors. First, you will learn how to read molecular structures from smiles and understand what information they do and do not encode. Then we will cover stereochemistry and why small differences in three-dimensional structure can have large biological consequences. And then we will introduce a set of core descriptors: logP, logD, pKa, TPSA, and hydrogen bond donors and acceptors. You will see how specific functional groups give rise to these descriptors. And finally, we will connect these molecular properties to binding, delivery, stability, and safety. So let's see some examples of these tools. Um, Before we go into individual descriptors, here is the key idea of all of these tools are Decade, OpenEye, Epic, whatever, right? So they are performing chemical interpretation behind the scenes. When you compute quantities like logP, TPSA, or pKa, the software is identifying functional groups, assigning charge states, and applying chemical rules to the molecular structure. Those calculated values are then used as numerical inputs for downstream analysis and models. So if you understand what these descriptors physically and chemically represent, you can recognize bad inputs, explain unexpected results, and reason about why a prediction behaves the way it does. So that's why. So this module is important. Molecular structure and how we represent it underlies every quantitative approach to drug design. So we can start with this is smiles input, and then from there we will move on. So smiles is simply the text format we use to represent molecular structures. What it encodes is connectivity, but not the molecule's 3D shape or conformation. So the same molecule can be written as many different equally valid smile strings. Canonicalization helps enforce consistency, but it is important to remember that canonical smiles are tool specific, not chemically universal. Stereochemistry is also not included by default. Only isomeric smiles using symbols like at or double at explicitly encode configuration. And even then, they still do not capture 3D conformation or environment. So this text string becomes the starting point for modeling. But chemically, it's a simplified two-dimensional representation. Next, we will talk about stereochemistry, when 3D shape determines biology. Stereochemistry is where two molecules can look identical in two dimensions, yet behave very differently in biological settings. Here is a simple example. L-alanine is the form used in human proteins, for example. Whereas D-alanine is its mirror image and plays a specific role in bacterial cell walls. So, in smiles, the only difference between these two molecules is the use of et versus doublet to encode configuration. Without these stereochemical tags, both collapse into the same 2D representation. If stereochemistry is missing, a structure-based analysis cannot distinguish between them, even though their biological roles are completely different. So that's why, before generating descriptors, stereochemistry must be present, verified, and consistently represented. Now let's see functional groups, the source of descriptors. Functional groups are the structural elements that give rise to molecular descriptors. Each functional group contributes characteristic chemical behavior, acidity, basicity, polarity, hydrophobicity. Acids and bases determine ionization and charge state, which is captured by pKa. Polar groups such as hydroxyls and amides govern hydrogen bonding and polar surface area. Aromatic rings contribute to hydrophobic character and influence logP. If you understand the functional groups present in a molecule, you understand where the descriptors come from and how molecular structure is translated into these quantitative properties. So let's talk about some core descriptors. So after parsing the smiles and identifying functional groups, we arrive at a set of core molecular descriptors. Log P and log D describe lipophilicity, which strongly influences both membrane permeability and aqueous solubility. pKa characterizes ionization behavior and determines how a molecule's charge state shifts across different pH environments, such as stomach and bloodstream. Um, TPSA, or topological polar surface area, reflects the extent of polar surface and is a key factor in setting absorption limits. And hydrogen bond donors and acceptors quantify a molecule's capacity to form hydrogen bonds, which affects both binding interactions and solubility. Finally, the number of rotatable bonds provides a simple measure of molecular flexibility and conformational freedom. Together, these properties form a compact chemical summary that underlies many empirical rules and quantitative analysis used in drug design. So let's start with hydrophobicity. Um, Water forms an extensive hydrogen bond network with itself. Non-polar groups cannot participate in that network, which makes their presence in water energetically unfavorable. As a result, water tends to exclude non-polar groups, effectively driving them to associate with one another. So this is what we call, the hydrophobic effect. So this effect underlies membrane formation, contributes to protein folding, and strongly influences whether small molecules can cross biological barriers. Hydrophobicity describes how non-polar groups behave in water, while lipophilicity, quantified by logP and logD, measures how strongly a molecule prefers a lipid phase relative to water. Let's talk about P and log P. Okay, so when we talk about log P, we are describing how strongly a molecule partitions into a membrane-like organic phase relative to water. The partition coefficient P is defined as the ratio of a compound's concentration in an octanol versus water. And log P is simply the base 10 logarithm of that ratio. Because this partitioning behavior reflects lipophilicity, log P is widely used as a compact indicator of membrane permeability and aqueous solubility. In general, high higher log P values are associated with improved passive membrane crossing, but poorer aqueous solubility, while lower log P values favor solubility at the expense of permeability. So, for this reason, log P is one of the most widely used single number descriptors in ADME analysis. Structurally, hydrophobic features such as alkyl chains and aromatic rings tend to increase log P, whereas polar functional groups such as hydroxyls, amines, and carboxylates tend to decrease it. Now let's go to pKa. All right. So now let's cover this second of our core descriptors, that is pKa. This describes a molecule's ionization behavior and resulting charge state, which strongly influences its biological behavior. Uh, to understand pKa, it is important to distinguish it from pH. pH, it's an environmental variable. It's a property of the solution a molecule is in, not the molecule itself. Different compartments in the body span a wide range of pH values from this the highly acidic stomach around pH 1.5 to 3 to a very well regulated pH of blood at around 7.4. In contrast, pKa is a molecular constant. It is an intrinsic property of a functional group within a molecule, something like a molecular weight, for example. Formally, the pKa is the pH at which half of the molecules exist in the ionized form. A more intuitive way to think about it is, something like this. Um, pKa determines how a molecule responds to changes in environmental pH. The pKa itself does not change, but because of its value, a molecule with a pKa around 4 will be largely ionized in blood at pH 7.4 and predominantly neutral in the acidic environment of stomach. So this relationship between molecular pKa and environmental pH is a key determinant of drug absorption and distribution. And then we will see why this pKa matters in ADME modeling. All right, so we uh, just saw that the pKa determines a molecule's ionization behavior and the resulting charge state, which strongly influences solubility, permeability, and binding. Um, let's use a classic example here, aspirin. So aspirin is a weak carboxylic acid with a pKa of about 3.5. So now, let's follow the drug through the body. So you take it orally, and then aspirin enters the stomach where the pH is highly acidic, around 1.5 to 3 that we just saw in previous slide. So in this environment, the pH is below the pKa. Under these conditions, aspirin remains largely protonated and therefore neutral. And then neutral species are more membrane permeable, which allows aspirin to cross the gastric epithelium. The drug then enters the bloodstream where the pH is, around 7.4. Um, so now the pH is well above pKa. So aspirin becomes predominantly deprotonated and negatively charged. So in this ionized form, aspirin is much more water soluble, but much less. So it's far less able to cross lipid membranes. This shows us, why weak acids like aspirin are more membrane permeable in acidic environments, but become increasingly ionized and effectively trapped at physiological pH. What you hear this the ion trapping effect. So this shows us how the interaction uh, between molecular pKa and environmental pH shapes drug absorption and distribution. So now let's see D and log D. So this is the real world lipophilicity. So we know that many drug molecules exist in both the neutral and charged forms depending on pH. Log P describes partitioning of the neutral form only, shown here as the flat gray line, this one, so which remains constant regardless of pH. For but for ionizable compounds, so this can be actually misleading. Log D, the distribution coefficient, extends this concept to real biological conditions. It measures octanol water partitioning at specific pH, which accounts for both the neutral and ionized species, right, present at equilibrium. So that is, the blue curve you see. Um, and then actually you notice what happens here at a low pH, uh, around 2 in stomach. Log D is high, around 3, because the molecule is mostly neutral and membrane permeable. But as pH increases, uh, you can see here, now you will see crossing 4.5, past pKa 4.5, ionization increases, and we see this ionization driven drop in the lipophilicity. By the time we reach around pH 7.4, that's the blood pH, log D has dropped to just like zero point 0.1 or something like that. So here, the molecule is now mostly ionized and has low membrane permeability. So this is why for ionizable molecules, log D at physiological pH, that's around 7.4, right, is the relevant descriptor for our modeling. Using log P alone can be misleading because it does not capture this pH dependent behavior. Now let's see about polarity and then also size. Okay, so uh, two properties primarily govern a molecule's ability to cross membranes, right? That's size and the polarity. Size, it's just straightforward concept, that as molecular weight increases, passive diffusion across membranes generally becomes less favorable. Polarity arises from these hydrogen bond donors and acceptors, and is summarized by this total polar surface area, TPSA. As polarity increases through higher TPSA or more hydrogen bonding functionality, uh, passive membrane permeability typically decreases. So you can see an example here, that is the atenolol metoprolol comparison actually illustrates this clearly. So these two molecules have nearly identical molecular weight. You can see here, 266, 267, but very different polar surface areas, leading dramatically different abilities to cross membranes. See, um, and then see here you can compare uh, so these molecules. So this is low passive permeability, whereas here high passive permeability. So this can cross membrane a bit easily. Now finally, let's see summary for this lecture. So we started with the smiles as a standardized way to encode molecular structure, and why canonicalization and stereochemistry are essential for producing consistent and chemically meaningful inputs. From there, I think we examined uh, core descriptors that emerge from functional groups, pKa, logP, logD, TPSA, those things. LogP captures the intrinsic lipophilicity of the neutral form, while logD extends that concept to a specific pH, accounting for ionized species. I mean, which is actually what matters most in biological systems. pKa determines how a molecule's charge shifts across environments, and TPSA, HBD, HPA describe the polarity that governs membrane permeability. So together, these descriptors form the chemical foundation for understanding absorption, distribution, and binding. And they are the inputs on which more complex modeling approaches are built. I mean, we need to understand this, right? These descriptors don't determine outcomes by themselves. They are, correlated features that models learn from, and their apparent importance depends heavily on chemical context and a dataset bias. Thank you.